This is Stanford Engineering's The Future of Everything, and I'm your host, Russ Altman. There are so many neurological diseases characterized by dysfunction of the motor or sensory systems in the brain. Parkinson's disease manifests as tremor, rigidity, but also has sensory components like the loss of taste. Neuroscientists have discovered that part of the problem in Parkinson's disease, and probably other diseases, is an abnormal synchronization of the neurons that lead to decreased diversity in the outputs of the system, in many ways leading to reduced flexibility in the brain. Invasive techniques, such as deep brain stimulation, can often break this abnormal synchronization to desynchronize cells and improve symptoms. Other diseases also have this hallmark of excessive synchronization, and they include tremor, epilepsy, muscle rigidity after stroke, and even some psychiatric diseases like obsessive-compulsive disease and major depression. Amazingly, desynchronization can be achieved without implants or electrodes, but simply with vibrations delivered to the fingers in a carefully designed manner. Professor Peter Tass is a professor of neurosurgery at Stanford University. He has developed a glove that administers light vibrations to the fingers, and it can improve the symptoms of some diseases in a matter of weeks. So Peter, you're a neurosurgeon, and when many people think about neurosurgery, they think about an open brain and scalpels and carefully dissecting brain tissue. But you have done research on something that's an entirely different approach. Can you tell me what that approach is and why you've spent so much time trying to refine it?
Yeah, well, just to avoid misunderstandings, I'm not a practicing neurosurgeon. I'm a professor in neurosurgery. It's a great department. We have a couple of really first-rate basic scientists. I'm a translational scientist. I studied I'm a medical doctor, physiologist, mathematician, and physicist. And the goal of what I'm doing is to design, by means of computational studies and maths and physics, to design simulation techniques that enable to reshape neuronal networks in a way that they unlearn abnormal activity in order to induce long-term therapeutic effects.
And what are some of the diseases that you have in your sights initially that you think are good candidates for this? And then I really do want to get into the underlying physiology of what's going on. But first, for the applications, what are some of the promises of these technologies?
Well, there are a couple of disorders. Several brain disorders are characterized by abnormal neuronal synchrony. Classic one is Parkinson's, essential tremor, dystonia, epilepsy, stroke, but also there are also other neuropsychiatric disorders, for example, OCD, obsessive-compulsive disorders. There's a wide range of possibilities.
And now tell me, what are the interventions? Because I think people will be surprised that this is not something that you might think of as surgery, but it's still, nonetheless, it's an intervention. So can you describe the types of interventions that you take in addressing these diseases?
Yeah, well, the first step, I've been working on this for a while, was to so to speak, improve deep brain stimulation by means of model-based, computationally developed stimulation patterns. And the next step now is to take this to a non-invasive level, because many patients are really scared by brain surgery. And the other thing is, it's also important to provide patients with therapeutic means early on. And whenever you use brain surgery, you have to wait, because there's an intrinsic risk since it's an invasive procedure, and we would like to provide patients with a therapeutic means that has a huge potential early on.
So tell me, one of the things you've written about is, I think it's called Vibrotactile therapies or approaches. Tell me a little bit about what that is and how it works.
Well, the Vibrotactile stimulation, specifically Vibrotactile, so-called coordinated reset stimulation, means that you deliver vibratory bursts, really weak bursts. They're not painful at all. They're not distracting. You could have them on your fingertips now while not being perturbed while we talk. And these vibratory bursts are special in the sense that they control the firing of neurons in the thalamus, the sensory gateways in the brain, as well as in the cortex. And that's what we need. We need to control the timing, the discharges of the neurons in order to realign their discharge patterns, to make them unlearn abnormal neural connectivity. And in this way, although we apply them to the fingertips, these mild and weak stimuli, we can really change neuronal connectivity, neuronal firing in the center of the brain and in the cortex.
So how was this even discovered? So it sounds almost like magic that relatively mild vibration signals in my fingertips could essentially start to rewire my central nervous system. What was the basis for the discovery of this potential, and then how have we learned to use it in a rational way, in a focused way for specific disorders?
Well, there's one important point, and that is, so far we do not fully understand how classical standard deep brain stimulation works. The important thing about coordinated reset deep brain stimulation, so the invasive variant, is that we use phase resets of rhythmically active neurons, meaning when a neuron is actively written, we restart the rhythm of the neuron. In particular, we do not suppress the neurons, or we induce whatever type of abnormal firing rates or the like, but we just control the timing of the neurons. And there are beautiful studies by Lance's group that have shown in humans, in patients, during depth electrode implantations, and that they've shown the following. During a vibration cycle, so when you vibrate the skin during one vibration cycle, you have a beautiful, what's called phase locking of the neurons. In other words, the neuron cares about this cycle, vibration cycle, meaning if you plot the neuronal firing over a vibration cycle, it's not a uniform type of diagram, but there are peaks. So in other words, we know that we can control the timing of the neurons, and that's what we need. We do not need, and we do not want to suppress the neural firing, because neurons have to be active. They have to be active in order to learn and unlearn their connectivity patterns. And the most important thing is that we can move them into a certain dynamic state, desynchronized state, and that's possible by delivering these vibratory stimuli at different sites, meaning different fingertips, at different times.
So so I have so many questions. I'll try to do this in an orderly manner. So first of all, obviously vibration is a sensation, and so you're hitting sensory neurons. We know that sensory neurons have connections with motor neurons, the things that make things move. How deep into the central nervous system can you get, however, if your port of entree is these sensory neurons? Will they allow you to reach, so to speak, deep into the brain, or is there natural boundaries because you're mostly entering through the sensory system?
No, you can reach as deeply as you can by means of a depth electrode, by means of an implanted electrode. You reach the sensory thalamus. So it's in the midbrain. It's an important nucleus in the midbrain, and from there it goes up to the cortex. And the cortex is central, most probably also for the effect of even standard deep brain stimulation.
And the other question is, you've referred to fingertips, and I think a moment ago you even said that it may be different fingertips that you differentially stimulate with these vibration. What is special about the fingertips, and are there other parts of the body that give access to different parts of the brain, perhaps, or is there something special about the hands?
One could use also skin of different parts of the body to start with. We started with the fingertips simply because the fingertips have a huge cortical representation. When you think of the so-called homunculus, the homunculus is a being that has huge hands, in particular huge fingertips, and that means there are many, many, many neurons that are responsible for the sense of touch. That's how we are built. That's important to us. And therefore, what it means to stimulate the fingertips means we have a large neural population. And by stimulating only a little bit of the skin, meaning the fingertips, we already reach a relevant part of this neural population. And that's the elegance, in some sense, of it. And what we know also from many studies, computational studies, but also proof of concept studies with CR deep brain stimulation, is that desynchronizing effects propagate within the nervous system. So it's not that we have to stimulate, for example, the hand, if there's let's say a hand tremor, or the foot, when there's a foot tremor or whatever. But these effects propagate, and therefore, amazingly, it's sufficient to use just one entry point, and then the effects appear to propagate through the entire brain, or to a large part of the brain.
Gotcha. Okay, so this is quite exciting, and I know one of your areas of initial focus is Parkinson's disease. So for people who don't think about Parkinson's disease, could you characterize for us what is the deficit, and then what is the opportunity for this desynchronization to help either the symptoms or the progress of the disease?
Yeah, Parkinson's disease, what's most known about, or typically known about Parkinson's disease is the motor deficit. Tremor, rigidity, and bradykinesia. Tremor means it's clear it's shaking, rigidity is the stiffness of the muscles, and bradykinesia means the slowness and the inability to properly initiate movements. However, Parkinson's is way more than just a motor impairment. Parkinson patients also have substantial sensory impairments. For example, they have enlarged receptive fields. A receptive field tells you, for example, if you look at this particular vibratory mechanoreceptor receptor that picks up vibrations, then, for example, in a normal subject, this receptor would get activated by vibrations up to here, for example, but in a Parkinson patient might even be up to here.
So you're pointing to your finger, for those who are not watching, you're pointing to your fingertip and saying that in Parkinson's, a vibration stimulus at the tip of the finger might even fire or affect sensors that are in the middle of the hand.
Yeah, that's the point. And Parkinson, and for example, what does it mean in everyday life? For example, we had a Parkinson patient who told us that at the beginning, prior to our study, he wasn't able to tell what kind of coins he had in his pocket. But after 3 months of treatment, he was able to differentiate the different coins. And the other thing, it's not just sensory impairment, since, the brain is hugely connected, and the different systems are highly connected, and purposefully from a biological standpoint, and also the so-called sensory motor integration is impaired in Parkinson's patients. It's always important that when you act, you know that you have the right feedback in order to plan, correct your actions, and so on.
So tell me now, okay, so that gives us a good introduction to Parkinson's disease. What is the underlying deficit that the vibration therapies can kind of undo? What deficit can we identify and then try to rectify with these vibration signals from the hand?
Yeah, so Parkinson's is a multi-system degeneration. It's by far not restricted to the midbrain, to, for example, the substantia nigra and other basal ganglia. But it's a multi-system degeneration that starts, for example, in the olfactory bulb, where patients do have smell loss, years, for example, before the motor symptoms occur, because only when there's about 50 or 60% of the neurons of the dopamine-producing neurons that die, only as soon as this happens, the motor symptoms occur. Now, the important point now is to a lack of dopamine is that there's a cascade of, so to speak, probably in some sense a vicious cycle type of cascade of plasticity-mediated processes, which lead to abnormal synchrony, abnormal neuronal synchrony, and abnormal connectivity.
Okay.
And what we do is we specifically counteract abnormal neuronal synchrony by desynchronization. You can do lots of things to abnormally synchronized neurons. You can suppress them, inhibit them, or whatever. Or you can force them to act in a different rhythm or something like that. What we do is we desynchronize them. And the important point is, if you do it in the right way, and that's what we're aiming at, is that the neurons unlearn their abnormal neuronal synchrony. And that's, for example, what we've shown in a pilot study. When we measured the EEG, when we recorded the EEG, meaning the brain activity in our Parkinson's patients prior to the glove treatment and after 3 months of the glove treatment off medication, to have them in a, so to speak, medication-naked state, we've seen that there was a huge decrease of the Parkinson's-related synchrony in the somatosensory cortex.
Okay, great. So that really helps. So there is a synchronization of the neurons that is abnormal, and the vibration therapy, and I want to ask you about the details of it because you mentioned the word glove, and now a picture is starting to form in my head. But with that therapy, you can undo that abnormal synchrony and give the neurons a chance to relearn what we might call normal interactions. So tell me, what is the therapy like for the patients? What are they wearing? How long does it last? Are there side effects?
Well, it's a so-called open glove, so it's not like a glove you wear for skiing or so, but it's open. There's enough or as much space for the skin to have normal physiological contact with the surroundings. And then at the fingertips, there are so-called tactors, mechanical stimulators that are mounted by means of Velcros. And these fingertip stimulators get activated according to a specific algorithm, a specific pattern. So typically, the two fingertips are not activated at the same time, or the two fingertips of the same hand. And this sequence of activation is based on theory, is based on computational research, and aims at optimally desynchronizing abnormal neuronal synchrony. And concerning the side effects, fortunately, we haven't observed side effects so far.
And how long do they wear these gloves?
In our studies, we had patients wearing them 2 hours a day or mainly 4 hours a day. The point is, probably it's not necessary to do this for a longer period of time, so the required dosage will certainly decrease, so it might well be based on also case studies we performed. It might well be that, for example, first 4 to 6 weeks, 2 times 2 hours, in total 4 hours a day, should be optimal, and then the patient can go down to 2 hours, and then after a while to only 2 to 3 to maintenance dose of something like 2 to 3 hours per week.
This is The Future of Everything with Russ Altman. More with Peter Tass next on SiriusXM Business Radio Channel 132. Welcome back to The Future of Everything. I'm Russ Altman. I'm speaking with Professor Peter Tass of Stanford University. In the last segment, Peter discussed how vibrations applied to the fingertips can reach back all the way into the brain to reset abnormal circuits and allow them to normalize. In the next part of our conversation, he describes how he got interested in the ways in which systems, like cells in the brain, organize themselves, and how these organizations can become exquisitely sensitive to small changes. And that's the key for this vibration therapy. Those small changes are what he is introducing with the vibration therapy, and he'll describe several other potential applications coming down the pike.
During my medical studies, I got really intrigued by self-organization processes, although at that time I didn't even know the term. So I was intrigued
we might need some help about what you mean by self-organization.
Yeah, I was really intrigued by how many subsystems and parts of the body work together in such a well-coordinated, well-orchestrated way, both under healthy but also at normal conditions. It's amazing in some sense. And I thought at that time, well, I don't learn the methods I need in order to properly understand and potentially manipulate or modulate this. And that's why I also studied maths and physics. Fortunately, had the opportunity to study all the relevant things. For example, my PhD supervisor was Herman Harten, the founder of synergetics, which is a self-organization theory. The important point about self-organization, if you think of patterns like clouds in the sky, how do they form? One important message of self-organization is tiny things and even unspecific things may change the collective behavior of a large system massively. So in other words, we do not need to massively interfere, but it's sufficient to do this in hopefully the most intelligent and weak and gentle way. But nevertheless, we can induce massive changes. We have to trust in a way, you see. It's in some sense, we have to trust that the brain acts as it is, as it's acting. We use the same principle, learning principle, that, for example, somebody uses when he wants to learn, or she learns to learn playing piano on an instrument. That's the same mechanisms we use for our Parkinson's treatment.
So that's fascinating, and it leads to questions. do you think that this vibration, the effectiveness of the vibration, does that indicate that evolutionarily humans may have feedback loops with the outside world that kind of maintain homeostasis, maintain balance, that we might not be aware of? Because the fact that this loop even exists, that with the vibration you can send signals to my brain to synchronize, desynchronize, makes me wonder if over evolutionary time this has had actual advantages to the organism out in the wild. Has there been any suggestion that this might be part of our adaptation to life on Earth?
Well, I think it's a very good question in the sense, in the following sense. I think the brain, and we as a whole, so to speak, the body, but more than even the body probably, we have to fight for our homeostasis, for our normal functioning. And there are many processes underway under healthy but also under normal conditions to keep up the current state. And for example, what we are currently doing in our computational modeling, we take into account homeostatic principles, simply because otherwise would not be able to describe particular phenomena that we've seen. For example, we've developed a tinnitus treatment. It's the same principle, but instead of vibrations, we use tones.
This is for ringing in the ear.
Ringing in the ear. Tinnitus, yeah, ringing in the ear. And what we've seen there is the following. And homeostasis, and in other words, what's also called structural plasticity. Structural plasticity means you really terminate, you stop, you prune neuronal connections. You do not just weaken them, but they really vanish, or you newly generate connections. It's an even more, let's say, massive type of mechanism that the nervous system uses, because the nervous system only maintains, it's all about metabolism in some sense, but also space, only maintains connections that make sense from a biological aspect. And what we've seen for example, in our tinnitus patients is the following. So after about 3 months, so 10 to 12 weeks treatment, they're doing a large number, 80% or more, are really doing well. And under real world conditions, in other words, after the study, when this thing got FDA and CE mark approved, patients didn't stimulate that much anymore. However, when then, for example, something really horrible happened, they lost their job, somebody died or whatever, subjectively they had the impression tinnitus is back as it was before. However, when they then took on the stimulator, it was gone. That's what a couple of patients described me within only 2 hours. So there's a memory effect, you see. And homeostasis is really so super important for really fighting abnormal, for example, activity. And that's something we are currently working on. It's a hot topic.
So I'm excited to hear, first of all, you mentioned the FDA, so it sounds like some of these therapies are already available for patients and their physicians to use. Is that true?
Yeah, the tinnitus treatment, yes, and currently we are running an FDA approval trial for the Parkinson's for the glove treatment.
Very, very exciting. So let me ask, in our final kind of 2 or 3 minutes, what are some of the other neurological diseases, and I guess psychiatric perhaps, that you think are within the range of these new technologies? Because it's really remarkable in terms of, you said the side effect profile is very low. It's not it's not at all invasive. So where do you see other applications arising?
A very natural one is essential tremor, and we treated Parkinson patients who had pre-existing essential tremor, and they had a huge benefit for the essential tremor too. So that's one thing. Essential tremor is also characterized by abnormal neuronal synchrony. Another one is dystonia. Dystonia is in some sense a perfect target because of the abnormal activity. You see there's one, and of course also epilepsy, but there's one common, in a way, feature of dystonia, essential tremor, Parkinson's, and that is neurons. Why is synchrony not good? Synchrony intuitively sounds nice. Right. Neurons do something together in a perfect way and so on. That's not good. Simply not good, because neurons should be free to process different aspects of information processing, so that we can move, for example, the index finger separately from the pinky and so on. But if everybody is synchronously active, we're no longer able to do this. You see, it's important to be selectively free to selectively process information for this. So as I said, and also epilepsy, we've been starting with this together with Bob Fisher here at Stanford and Baba Grasavi. And on the neuropsychiatric side, I think OCD would be a fantastic application, and we are preparing for this.
When you approach a new disease, how do you figure out what the right protocol should be for achieving the desynchrony? Is it that desynchronization is a general phenomenon that you can apply without specificity, or do you have to have specific types of desynchronization for different disease states?
Well, you have to adapt it, and to some extent to the specific physiology. The main principles are quite general, but you have to adapt it to the specifics. I mean, for example, the frequency ranges are different for the different brain disorders, but nonetheless, it's quite general.
And so there's an interaction between the natural frequencies of the brain in each disease state and how you might elect to do the vibration stimuli in order to kind of match the frequency ranges.
Yeah.
Fantastic. I guess the final question is, how can people who want to learn more about this? Because I think it's somewhat stunning. It is not in the public consciousness currently, and so where should people look to learn more about the growth of this techniques?
Oh, well, they could, for example, check my lab's homepage.
Peter Tass?
Yeah, exactly.
At Stanford University.
Exactly, yeah.
Thanks to Peter Tass, that was the future of neuromodulation. You have been listening to The Future of Everything with Russ Altman on SiriusXM Business Radio, Channel 132.